Okay, so this is the final chapter for this class, chapter 23, right? And it basically talks about how metabolism is integrated in a very complex yet very organized mammalian biology. And so the question is, how is metabolism integrated in complex organisms that have multiple organ systems, right? And when we talk about organization of metabolism, we are really talking about synthesis of new proteins, new biomolecules, or degradation of those biomolecules. How are they coordinated among all these organs? And so it turns out that in a complex organisms like us, we have evolved a very, very interesting system where hormones try to integrate and coordinate the activities of different tissues. And they optimize allocation of fuels and precursors if they are needed for biosynthesis to each organ. Okay, and so that makes sense. But the beauty of integration is really, if you look at the complexity of the system, you will actually appreciate how well the whole thing of both catabolism and anabolism is integrated. Right, so organ systems in us, mammalian biology, for example, they have very specific roles, specific functions. Brain, for example, is very, very involved in responding to external stimuli, regulating things, thinking, fight or flight type of activities, right? And so brain likes to utilize glucose as its primary fuel. Glycolysis can actually rapidly produce ATP. So there are some special functions to special organs, okay? And some organs actually play a sacrificial role in ensuring that other organs are not deprived of resources. For example, liver plays a very, very important role in utilizing readily available fatty acid as the energy source. It does use glycolysis, not to say that it doesn't, but it preferentially utilizes fatty acid as fuel. And so glucose is readily available. Remember, liver also does gluconeogenesis. And therefore, it makes new glucose molecules for the organs that actually primarily rely on glycolysis for their energy production. So you can see the specific roles these specific organs play. You already know that major fuel in us are glycogen, right? Glycogen stored in the liver and muscles. And then the neutral fat, triacylglycerols, stored in adipose tissues. And we don't call protein as, storage fuel, but there are some proteins that are there in skeletal muscles, and they can be utilized sometimes when the cells are starving. For example, in type 2 diabetes, remember? We talked about that. But, there is a usual order of preference for use of these fuel. Usual is in quote, okay? Glycogen is first, then triacylglycerols, and then protein. Okay. And so if you remember, glycogen levels drops between the meals in the liver, right? Because liver is trying to ensure that the sugar levels are maintained throughout constant, even when you are hungry, 5.5 millimolar, right, in the blood. But it's in quote because you know that liver primarily utilizes fat, triacylglycerols type of molecules for fuel. Okay, so that's something that you have to just keep a little bit in context. Cool. So with that, how is metabolism coordinated in mammals? It turns out that it is primarily achieved by this neuroendocrine system. Neuroendocrine system consists of, neuronal signaling as well as the endocrine signaling. Okay, and that is what is shown here on the top right hand side. In the neuronal signalings, you can see they are showing you a neuron, right? Neurons will produce nerve impulses like electrical signals that will originate from the cell body of the neuron in the center somewhere here, but they can travel, those electrical impulses can travel very rapidly over long distances to the tip of the axons here. Okay, where once they travel, that electrical impulse will end up, in the output where axons will release neurotransmitters, for example, acetylcholine, right, or GABA. Those neurotransmitters such as acetylcholine are released, and then they will diffuse to their target cells, shown here. Target cell can be another neuron, right? Or it could be a muscle cell, some myocyte, or a cell that actually will release certain hormones in that response of the neurotransmitter. And they're typically very close by, a fraction of a micron, or maybe at the maximum a couple micron away. Okay, so the neurotransmitter rapidly goes and binds to the receptor on these neighboring cells. It doesn't diffuse too far, right? Within a few micron. That's how neuronal signaling works. In the endocrine signaling, hormones are typically released as secreted into the bloodstream. Okay, so you can see there are two different types of cells. They're secreting hormones, as shown here in the blue triangle and red rectangle. They're secreted into the bloodstream, which then will take them throughout the body, right, to the target tissues. And those target tissues could be really far apart, meters away, right? They are far away from the secreting cells. And so both of these, right, neurotransmitter and hormones, they go on and interact with their target cell receptors and elicit biological responses, whatever that could be. Okay, if muscle has to contract, it will contract if it's a muscle cells, right? So that's the neuroendocrine system. Now I want to show you the structure of a hormone called Thyrotropin-Releasing hormone, TRH, here. If you look at this molecule, it basically is a tripeptide hormone, but in a modified form. It's a tripeptide made up of glutamate, histidine, and proline, but it has modification. You see how they are showing NH2 at the C terminus? It usually means that that C terminus is an amide. That's how they write peptides, if you recall, right? Convention. Pyroglue is where the glutamate side chain has formed an amide bond with the amino terminus. Cyclic, you can see here. So you have two amide bond in this TRH, thyrotropin-releasing hormone. Now, this hormone, there's a big story almost, to study in 1960, to study the effect of this hormone, right? Thyrotropin releasing hormone. 1.5 million pigs were slaughtered, and then their hypothalamus were collected, ground, and basically it was a really, really big effort to isolate this molecule so people can study what the function is. Okay. It's also known as TRF, right? It's a tropic tripeptide hormone, and it is released from hypothalamus. What is a tropic hormones? Tropic hormones are these hormones that have other endocrine glands as their ultimate target. Okay. So most tropic hormones are produced and secreted by the anterior pituitary and hypothalamus, just so you know. Okay. And once this hormone is released from hypothalamus, right, it will stimulate the release of thyroid stimulating hormone, and from anterior pituitary and other targets. And so thyroid hormones, I don't want to say too much, but thyroid hormones are very, very important, right? They are made by the thyroid gland, right? And two main types of thyroid glands, thyroid hormones are released. They are called T4 and T3, right? And so these hormones, affect every cells. Very, very important, actually. If you are not feeling well, sometimes your thyroid hormones might be off. And so you might want to tell your physician, let's say you are feeling extra fatigue, or your heart is, running too fast without any reason. Okay, so too much of thyroid hormones can speed things up, and too little will make you lethargic, will slow things down. Okay, so thyroid hormones play a very, very important role, in many things that people don't realize. It actually controls your metabolism. It regulates how body burns calories, right? If they are off, you can gain a lot of weight, or you can lose a lot of weight if it is hyperactive. It can slow down and speed up your heartbeat, right? It can lower or increase your body temperature. I mean, it affects many, many things, quite frankly, very, very important hormones. And there is a very interesting story that I usually share, but I will share that after the class, if we have time, if you remind me. But let's keep moving, okay? So if you actually look at this hormone, you will see that it's a very cool molecule. It's a very small tripeptide, but turns out that N-terminus residue and C-terminus residues are modified, right? You have this pyroglutamate and prolylamide. It turns out that this is very important. This modification that is done here is critical, so that this hormone goes and binds to its receptor only. Because remember, you can digest a lot of proteins, and you can find sometimes tripeptides like glu-his-pro from any other protein. But that glu-his-pro will not go and bind to the receptor. It has to be pyroglu and proamide. Why? You know this already. If it was not, this glutamate was regular glutamate, then its charge status will be very different, right? Glutamate side chain will lose its proton, amine will be protonated. Similarly, carboxy terminus will be deprotonated. It will carry one negative charge, right? Histidine also has an ionizable pH, right? pKa. So, this in a typical protein, let's say you had like 60 kilodalton protein, right? Charges on this amino and carboxy terminus contribute very little to the overall charge in the protein, because there are so many side chains. But on a small molecule like this, tripeptide, these two charges, dominate the property of this molecule, right? Basically, they remove the charge once you form this amide kind of structure. And that is very, very important in recognition, so that these hormones only go and bind to their receptors, because their receptors will not bind a tripeptide which does not have this kind of charge distribution. Cool? Very, very important. So this is a peptide hormone, small peptide hormone, TRH, just so you know. And it turns out that there are two general mechanisms of hormone action. Okay. There are these peptide and amine hormones. They typically bind to receptor on the outside of the cell, receptor that is already pre-existing. Okay. So in the plasma membrane, you'll have a receptor. Remember, insulin, for example, will go and bind to insulin receptor. That will be an example, right? Insulin is a peptide hormone. I'll show you the structure one more time today. Amine hormone will be an example, will be, for example, GABA, okay? Epinephrine. These will go and bind to a receptor that already exists on the membrane, plasma membrane. So they will not to enter the cell themselves. And so as a result, these peptide and amine hormones are much faster acting. Okay. Just seconds after this adrenal cortex, secretes epinephrine into the bloodstream, molecule shown here in the lower left, the skeletal muscles will respond, okay, right away. They will start breaking down the glycogen, like within seconds. Okay, that's how they're fast acting. They're typically water soluble, right? And they will alter the activity of pre-existing enzymes. That's why they're very fast. Their responses are quite fast. On the other hand, you have these hydrophobic hormones like steroids or thyroid hormones. They can enter the cells because they're hydrophobic. They can cross the plasma membrane and they will enter the nucleus, where they will go and bind to the transcription factor, etc. And they will alter the transcription of specific genes. And then as a result of that, you have altered amount of newly synthesized proteins, right? So they are very different than these peptide and amine hormones, right? These thyroid hormones or sex hormones, for example, they promote their response, maximal response to their target tissues, only after hours or days, right? So that's the two general mechanisms of hormone actions. And here they're actually showing you the classes of hormones. There are some examples here. You have seen some of these, right? Insulin, glucagon. They're peptidyl hormones. They act through plasma membrane receptors. Steroid hormones like testosterone, synthesized from cholesterol, they work through nuclear receptors. They typically regulate transcription, right? So steroid, vitamin D, retinoic acid, etc., thyroid hormone. And then you have a class of its own, nitric oxide is also a hormone. It's synthesized from arginine and molecular oxygen. Its receptor is a cytosolic receptor, guanylyl cyclase. Okay. So these are the classes of hormones, and you can see how they're synthesized and what their mode of actions are. There is a homework that I ask usually for you to go and look that up, the difference between endocrine, paracrine, and autocrine hormones. Now I will just tell you, and I will still ask you to go and look that up. Endocrine hormones are these hormones that are released into the blood, and then they are carried to the target cells throughout the body, right? Example will be insulin and glucagon released from pancreatic cells, right? Paracrine hormones are released into the extracellular space, and they typically diffuse to the neighboring target cells. Okay, they typically diffuse to the neighboring target cells. And icosanoid type of hormones will be, an example of paracrine cells, paracrine hormone. In autocrine hormones, as the name implies, they affect the same cells that release them. In fact, they will be released by the cell, and they will go and bind to their own receptor on the cell surface and regulate their actions. So those are called autocrine hormones. Okay. So let's look at one of the example, right, of endocrine hormone, insulin. You know that it's a peptide hormone. It's formed from a large precursor molecule, which is preproinsulin, right? I have shown you this slide, but I'm just reviewing this for you here. Preproinsulin is a single polypeptide comprised of an N-terminal signal sequence, right? That signal sequence has 23 amino acid segment, okay? And so when insulin needs to be released, that signal sequence is actually cleaved off. And then you generate what we call proinsulin. Proinsulin has 3 disulfide bridges, okay? So there you can clearly see they're color coded you A chain, B chain, and C chain, right? So proinsulin has still A, B, and C chains, and you can see that there are disulfide bridges, 3 disulfide bridges. And then further cleavage will actually clip the C peptide from the middle right here. So now C peptide has left, and your mature insulin has only A chain and B chain with 2 amino terminus and 2 C terminus and 3 of those disulfide bridges. That mature insulin will have the right shape to go and bind to the insulin receptor and start the signaling process, right? So here is the major endocrine glands that I'm showing you. They're color coded in pink, right? You have hypothalamus, you have pituitary, right, gland. Then you have thyroid gland. As I said, thyroid gland is very, very important. Lots of very, very crucial function in metabolism. Then you have parathyroid glands right behind the thyroid glands, right? Parathyroid glands are very, very important. They exist in your neck behind this thyroid gland, and they help your body control the levels of calcium in the blood. Very, very important. Adipose tissues, they act as a gland. They also secrete hormones like leptin. Okay. Adrenal gland that sits on the top of the kidney, right? Adrenal cortex. That's a very important, adrenaline is released from adrenal gland. You know that. Pancreas is, very, very important. It sits in the upper abdomen, right? And basically serves as 2 glands in one. It really is a digestive exocrine gland and is also an endocrine gland that produces hormones and secrete them. Very, very important. Both exocrine and endocrine gland, pancreas, right? So functioning as an exocrine gland, what will it do? It will secrete enzymes that will break down proteins, sugars, right? Lipids, fat, nucleic acids, etc. And when it is acting as an endocrine gland, then it will secrete hormones like insulin, like glucagon, right? That will control the blood sugar levels throughout the blood. Kidneys also play a role in endocrine signaling. Then you have this reproductive system, right? Ovaries or testes. So they are the major endocrine glands shown here in this figure. Now, how does the system works, and how, when the hormones are released or the stimuli is perceived, how the signal is communicated to the target tissues is what is shown here in this slide. Okay, and this is a very busy slide, but I want you to get the big picture of this, right? Hypothalamus is the central coordination system of the endocrine system. Okay, and hypothalamus directly talks to anterior pituitary and posterior pituitary through specialized connections. So any sensory input from the environment, right? Could it be threat? Could it be hunger? Whatever it is received by the central nervous system, right? Especially to hypothalamus. Hypothalamus, as I said, is the main center that coordinates the activity of this endocrine system. So based on whatever type of stimuli, it will produce what we call regulatory hormones or releasing factors. Okay, it's a cascade. Yeah, it's a very interesting. Signals that originates from the top, right, is passed via a series of relay mechanisms, literally, passing the baton to ultimate target tissues that are sitting at the bottom, right? And so you can see that these blue arrows are the connections between them, right? Direct connections, or if you see these dashed arrows, that dashed arrows represent the neuronal connections, electrical impulses. So hypothalamus, whenever it receives the signal, will release these releasing factors, also known as regulatory hormone, right? And so then they will go to their first targets. First target could be anterior pituitary. Now, anterior pituitary, has various types of hormones that it will release, right? They are showing you different types of hormones like corticotropins, molecular weight of 4,500, thyrotropin, molecular weight of 28,000, right? So different hormones are released by the anterior pituitary, and these different hormones released by the anterior pituitary have different secondary targets, right? So the secondary target could be adrenal cortex for ACTH. For thyrotropin, it could be thyroid, right? For these other hormones, it could be ovaries and testes. So those second target tissues will then release another sets of hormones here, right? So you can see that adrenal cortex could release cortisol or corticosterones or aldosterones. Thyroid glands will release either T4 or T3 or both. Ovaries or testes will release sex hormones, and they will eventually go to different target tissues, their last tissues. Okay. And so, different hormones have different targets. For example, if it's a liver cell, right, one of the function of cortisol in the liver cells, hepatocyte, is to increase the rate of gluconeogenesis. So when cortisol is released and goes and binds to the receptors of a hepatocyte, it will promote the rate of gluconeogenesis. And it will also promote glycogen breakdown in the liver and skeletal muscles, right? Aldosterone, right here on the extreme left, bottom right, that is a hormone that you can see is released by adrenal glands, right? And it helps maintain blood pressure. It regulates blood pressure. Okay, aldosterone can increase the reabsorption of sodium ion and water. Okay, and it actually promotes release of potassium ion in the kidneys. And by doing that, it actually raises the blood pressure, aldosterone. Right, so you can see different hormones can have different effect on their target tissues. That's what I'm trying to get across to you. There is a hormone here that is released by the posterior pituitary when the hypothalamus is stimulated, known as vasopressin. Vasopressin is an antidiuretic, which means it will help absorb water. Okay, vasopressin is found in most mammals, right? It has two primary functions. One is to retain water in the body, and the second one is to constrict the blood vessels. Okay, so this is how the coordination happens. Hormonal coordination happens between the hormones and their target tissues. Now, if I want to actually convince you some more, right? I'm showing you two hormones here, oxytocin and vasopressin. Oxytocin and vasopressin see very similar molecular weight, but their functions are very different. Okay. If you look at the structure of oxytocin and vasopressin shown here, you will see that they are almost identical peptide, except that only two amino acids are different that are shown here in the pink. In human oxytocin, one of this is isoleucine. In human vasopressin, it's phenylalanine. In human oxytocin, one amino acid is leucine. The other one is arginine, human vasopressin. Just think about it. Just think how minor the differences are, but how important those differences are, that human oxytocin has a complete different function and human vasopressin has completely different function, which means they go and bind to their respective receptors only, right? Their carboxy terminus is an amide, right? It's a glycine amide, right? Oxytocin, you know the function of oxytocin, what that does? It basically acts on the smooth muscles of the uterus and mammary gland, right? And it causes uterine contractions during childbirth and also promotes release of milk, when the child is born. So during lactation process, this hormone helps out. Vasopressin, when it is released, it will increase the water reabsorption in the kidney, and thereby it will promote the constriction of blood vessel and it will increase blood pressure. Think about it. Similar looking in chemical structures with some minor differences. They both are released from the posterior pituitary gland, but they have completely different biological effects. You can just look at this slide and think how chemistry is so cool, how biochemistry is what determines the overall biological signaling and symptoms and physiology. How chemistry manages this so well, right? And so here, again showing you the cascade of hormone release that follows when central nervous system is stimulated, right? When the central nervous system is stimulated, like for example, hypothalamus will release the releasing factors. One such releasing factor is CRH, corticotropin-releasing hormones. That amount of that hormone CRH is in nanogram. Okay, very small amount of CRH is released by the hypothalamus. Then that CRH will go and bind to the anterior pituitary, and that will release the next set of hormones in microgram amount. which is ACTH here, shown here. And then eventually ACTH will go and bind to the adrenal gland, and adrenal gland will release the milligram of cortisol into the blood. And they can be released into the blood where they can go to their target tissues and do different function. If too much cortisol is produced, it will go and bind to the hypothalamus or anterior pituitary and provide a negative feedback loop so that the signal is stopped. Further release of cortisol is stopped, right? So you can see that the cascade is sensitive to regulation at several levels here, right? Through feedback inhibition by, in this case, ultimate hormone, cortisol. So cortisol regulates its own production in some sense, right? By the feedback inhibition of the biosynthesis pathway within a single cell. How cool is that? And that's where I think I should shut up or stop. Let me stop the recording.